Sorry, I have a sore throat, so my voice is not very clear. So thank you very much, Neil, and all the organizers to invite me here to share my experience at the interface between computational science and experimental science. I spent a fair amount of time in the laboratory to learn how to perform the experiments. And the reason is because when I was looking at this data at the beginning, I couldn't understand a single thing of what was going on. So I thought it was me wrong, but actually I went to the benches and I realized that there was a lot much more that I had to learn, and there was a lot of noise, so to speak, that needed to be handled. So it doesn't work. So what I'm going to do today, I want to give you a little sort of wrap up of where we started, where we are now, and where we are going in the future. And what are really the type of challenging that we face today with the data that we collect. I would like to do this from the aspect of a computational science who's worked with data and on the benches as well, and help you to understand what really are the complexity of this the complexity of this high throughput data, and what are the advantages and the disadvantages that it expresses. And I'm going to give you two examples, one of a clinical study in which I was involved from the beginning, and including actually the collection and the extraction of the RNA for the assays, as well as the analysis, and another one which I call past-present, because it's something in the past, but also it's still open. There is a lot of open questions that needs to be answered. And a new project that I'm very keen and I'm very sort of passionate about, which is on stem cell. And tomorrow morning, Marcelo Rivolta is going to give you a talk on otic stem cells and what it took to him to create a tool that eventually can be implanted in the ears of deaf people. So the future directions are, I hope, more questions, and we can make discussion more than actually telling you things. So, no, this doesn't work. Once there was the darkness of the microscope room, and I can assure you it was pretty dark. The scientists in this period could only do one thing: probe one gene at the time, look down a microscope, and biological systems could only be analyzed by looking at a single part, one part at the time. Resources and time were not enough to do this, even to do it simultaneously. So basically, what we call system-level analysis today, it was theoretically possible, but practically impossible. Everything changed with the advent of the Human Genome Project, which was announced in 2000 and was completed in 2003. With the Human Genome Project, now we were able to read our DNA. We were able to understand what were the coding instructions in our DNA, and this enhanced many research questions and urged the need to be able to quantify all these genes that now we know where they were and we could read all at the same time. And that signed the explosion of the high throughput profiling of systems. And particularly the technology exploded. We had initially microarrays, which started with two-color arrays and then with Affymetrix. And then today we use the Illumina sequencer, the HiSeq, which is a second generation sequencer. And now there is a lot of effort trying to work on a third generation sequencer, which is molecular-based sequencing. In theory, should be enormously faster and enormously cheaper than the actual, which is still a little bit in its infancy. But what also is going to change, it's present, but it's definitely going to change our future, is the way that now we have, through this incredible machine called Orbitrap mass spectrometer, which is able to quantify proteins and metabolites in a very, very precise and fine way. So once we gained all this technology and all the data that this technology was able to give us, then we were in the situation where we could do system-level analysis. And we could start to think about integrating all the various aspects of these systems that was only studied in the microscope at one part of the time. And that's what we're actually trying to aim to do today, is to do functional association between what we call differentially expressed genes. But this, it's not enough, because we are studying systems that are made of complex population of these things that are cells. So in order for us to really achieve something that we can transfer into therapeutics or even personalized approach for our health, we need to understand the complexity of the tissues that we're working with. And what we are doing now is, and this will be certainly the future, is to look at what's happening inside the cells when a disease, when it's diseased, or simply when it's dysregulated by a biological process that can be caused by a disease. And this happening with, again, thanks to the technology with the microfluidics sorters that help us to literally separate the cultures in single cells. And then we can analyze their DNA and all the omics. So this omics enabled definitely the complexity of the biological phenomena may be elucidated clearly. But also, what it is important is that we are able to handle, we will be able, not yet, the composition of the complex tissues. For example, very important in cancer, when you have a hierarchy of cells, or in stem cells, because they are very heterogeneous cultures. So I thought, I have a mathematical background, I love definitions. So we're here talking about personalized health. So the first thing I thought, oh, well, let me see what is defined as personalized health. And this is what, I'm not going to read it out to you, you can see it. But one thing that really struck, and in fact I put it in italic, is this is a medical model. But you, by now, probably realize that it's not just medical. There is a lot more that enables this model to create a tailored product that is tailored to the individual patient. So my aims to try to achieve this tailoring to the individual patient is first to try to combine gene-level analysis and pathway-based methods so that I can generate a functional profile of functional modules that govern these processes. You must probably have heard of priors and Bayesian functions. So in theory, this would be my prior knowledge through a computational high. And then what I would like to do is to use the high-throughput data to build predictive models, not at the single part, but at system level, and only then trying to define some therapeutic intervention when I know I can predict once what happens to the system once it's perturbed. So I thought, I'm not a biologist, but I thought that this was quite important to show to you, because the data comes from these things. And it is important to understand what is the complexity that is embedded in the data. And very often, actually all the time, we do not try to dissect every single detail of this complex scheme because it would be intractable. But what we really want to try to achieve is to define these regulatory mechanisms in what we call molecular biology, because it's on single molecules that occur, for example, just for simplicity, in one single system, which is the cell. So we start from the DNA. The DNA is transcribed in mRNA. Then there are other things that happens, and this is the action of microRNAs up here and non-coding RNAs that actually do have some other important function. Then something happens and it's proteins. But once the proteins are formed, there are feedback again on the mRNA through the activity of these specific particular genes called transcription factors. But more, once the protein are formed, with the interaction with the environment or whatever is around the cell, epigenetics comes to play a role, and DNA methylation again goes and acts on the initial DNA. So it isn't really, there is a little bit an addition of complexity to it. Between the mRNA and the proteins, there is another step. So the mRNA from the cytoplasm goes into the ribosome, and only the ones that goes into the ribosome is translated. And now we have the technology and the biochemistry to actually look at the translatome. And what are we doing? It's going out of the screen. So this is some work that is happening. In fact, I wrote here now. Can't see it because it's out of the screen, that I'm doing in collaboration with a colleague in Sheffield Institute of Translational Neuroscience, Guilherme Utberger, and we are looking at what is the two components, the cytoplasmic transcription and the ribosomal transcription, compared with what we collect when we gather the total RNA of the cell. And what is surprising, the numbers are not important. What is surprising, there is a very small overlap in genes that are differentially expressed. So now this means, does it mean that we've done something wrong? This is sequencing, this is RNA sequencing. Is there something wrong? Or we are looking at things in a different time? And in fact, is this a snapshot at a single time, or there are different rates by which all this happens? And it is like this. And what the work that we're showing we are doing, so we don't know the answer to these questions, is are these increments of time or difference delay, if you wish, why all these things happen, have more weight than we thought they did. And therefore, the picture that we have up here, it's not what we thought from the beginning when we actually analyze RNA. So if we really want to do some therapeutical intervention, maybe we want to look at this, because we know that that is where the actual protein are going to be made. So all this, I'm showing this to you to sort of give you a feel of the amount of complexity that is in the data. And this is the snapshot of the data. For example, RNA that you sequence contains all this. And, or partially, mainly this part here. And already this is complex. And if you put this into a tissue or into a culture where there is heterogeneity, then things start to be really, really seriously complex. So the question would be, what do we do? Do we give up? No, we don't give up. Well, we have to make sure that we have appropriate experimental design and appropriate tools that enable us to measure all this uncertainty that there is in this system that we can't really quantify yet, despite the technology. So, you know, probably you've seen some of these pictures. So this is a graph taken from this paper in Science, which is 2011, and you can see that the data that we are acquiring is really is shooting. This is 2011, so imagine where it is up here. Despite the problems with the computational, the storage, the challenges, I believe, are in omics is the informatical case, is analyzed and summarized, but more importantly, is actually to interpret this finding in a complex system and in a system level based analysis. So there are three main questions that I think we have to ask ourselves every time we do this. First is, what is the value and the definition of raw data? Because now we can go to such fine details. What actually is raw? And how we can analyze this in an effective way, and how we interpret that. So today, we can collect all this data very easily, very, very well, with high accuracy, precision. We can put them together, we can stick on top of them some protein, but at the end, what really does give the accuracy of a system-level analysis is the quality of the data that has been analyzed. If you start the analysis of the data with a high-quality data, then your accuracy really goes very high. And to achieve this, you have to have a clear and really well-defined experimental design. You have to have appropriate assay for testing your question, and importantly, an appropriate way of processing the raw data. That's why I've done all this preamble, because I'm hoping that, so by really, which was a little bit the process that I did through my career, is to understanding how the data was produced would enable me to also understand what were the appropriate processing methods that I had to use to analyze the data. So we know that the tools we need are, we just said, optimal experimental design. We need to have some good mathematical models, and you guys have seen a lot of this. We need to have also statistical tools, because we want to do predictions. We want to be accurate and sensitive. But also, we want to have computational skills. You know, you heard Mike this morning and what we can do with softwares and how we can improve, because this is a large amount of data that needs quite a lot of computation. But also, and this is not to underestimate, what we want, we want some visualization tools. Because what we want to do out of this huge amount of data, we want to identify patterns. And we want to do this in a very clear way, so we can interpret it very quickly, especially if we are going to use this in medicine. So I'm going to present to you two studies. The first study is a clinical study that in a way brought together everything that I told you so far. As I said, I was in the lab, I extracted. The only thing that I didn't do in this study was to actually bleed the patients. I wasn't allowed. So this study was funded by, it's a clinical study done on patients, funded by this biomedical research unit called the Cardiovascular Biomedical Research Unit, which was led by Professor Crossman. This was funded by a government scheme called Best Research for Best Health. It was an infrastructure funding which would pay for the infrastructure, but also for the research that was going into this infrastructure. And the aims were to drive innovation in the prevention, diagnosis, and treatment of ill health. In our particular case, we were looking at a syndrome called acute coronary syndrome, basically a heart attack. And more important, this scheme was created to translate these advances for the benefit to the patient, so bench to bedside. What we studied in this biomedical research unit with this program was the genomics of the acute coronary syndrome. This was done basically embedded into the medics, together with the data coming from the transcriptional profiling of all this data. We also had lots of medical data, you know, full blood counts, and body mass, and all these other criteria that the patients were basically tested on or simply checked as a normal routine. And the idea is, through using the transcriptional activity and the data, the personal data of the patient, is to try to identify some risks that the individual would have for a recurrent event, because the recurrent events are the killers. Now, also there was an idea, this coming from the medical side, is that then we can do a recategorization of this myocardial infarction, because in the sense that then if we know what are the risks, if we know what are the individual medical parameters, we can personalize the therapy and not treat all of them at the same way, because they may be different and they may be reacting, their risk of recurrence is different. And the other important thing, which comes really more on the basic science part, it was to understand the role of inflammation, that no one knows what triggers the heart attack. So everyone knows how it happens, but no one knows what is the trigger for it to happen. So there was this idea that inflammation or predisposition to inflammation, to acute response, inflammatory response, would be a higher risk of generating the heart attack. So why are we studying cardiovascular disease? I think you guys know this, most likely, because it's the #1 killer, at least in UK. I've taken, you can't see really well, it doesn't matter, you don't really need to read the numbers, but what I'd like to point out, this is the death in women in the United Kingdom in 2012, and these are the men. This red part is the cardiovascular disease, which contains all the type of cardiovascular diseases. And this is all ages. And this is cancer. Right, we are a little bit better off than the men, but it's still pretty bad. But with people with age under 75, cancer really takes over. This is cancer and this is cardiovascular disease, which is telling us that cardiovascular disease is an old age, an aging disease. And this causes even more costs to the NHS. So the reason we're studying cardiovascular disease is because it's very large and affects economically, and also it has a heavy burden on our sort of health system. How does it happen? What you see here, it's an aorta post-mortem blocked completely by blood clot from the rupture of a plaque, of a fatty plaque. So now this plaque starts building in our artery from the age of 12. I was in shock when I learned that, but apparently so. Clearly, lifestyle, activity, how we eat, sort of helps to reduce the probability that this plaque suddenly explodes like a balloon. And the platelets respond to this by clotting, because they think that the blood is coming out. And this clot blocks the artery, and the necrosis in the heart happens, which is myocardial infarction. And if it is a large area, all the electrophysiology of the heart fails and the heart stops, and that's it. So in this study, we recruited 170 patients who came to A&E and with acute cardiac chest pain, not necessarily with a heart attack, but they were definitely having cardiac chest pain. And these were recruited within the 48 hours of the occurrence of this pain. Some of these patients had a stronger heart attack and a milder heart attack, and other just had the pain, called unstable angina. And we tested this we tested the presence of an enzyme called troponin in the blood. We also built a time course of these patients following the recovery from the day they were recruited, whether they had or not a heart attack, up to 1 year. We only analyzed it up to day 90 because resources and many other factors enabled us to go to day 365. These were the tests that we performed, and these were the sort of tools that we used to store the RNA. We also did a microRNA quantification to try to look at what it was the impact of the small RNAs in all this. You will see, and in the next slide, I'm going to tell you that out of these 170 patients, we only used 33 patients because they were the only 33 patients that actually came to each one of these visits. So, what are the problems that we have, that we face, the challenges that we face when we have data of this type? First of all, we're dealing with a very heterogeneous background. Lots of people coming from different population. And this clearly introduces problem with splice variants. Maybe there are different SNPs. Maybe they also express different type of isoforms that might have an effect. And that means that they might have a different predisposition to the disease. And they also are human beings that interact with the environment and therefore can have other factors on top of that that can enhance this heterogeneity. So, which means that you have a lot of variations in this data. They're not always linear, and it's not always possible to model them in a global form. Then you have the biggest problem of all, missing data points. So I told you we recruited 170 patients, but only 33 were actually completing the whole time course. So the problem is huge, not so much because you don't know what to do with it. You can use inference, you can impute, but then you enhance the complexity of the model even more, and therefore you need more computational time, more resources, your accuracy goes down, and definitely you do not have enough samples to do a proper inference of the missing data points. So the last point, which is not to underestimate, is that not always we have big enough sample size. For example, for a cardiovascular disease, that sample of 33 patients is less than nothing. If you think of how big is this disease and how many people are affected by this disease and the heterogeneity of the genetic background that is involved there. But then what you say, okay, great, let's get together many different areas, labs, people in the world. Okay, great, fine. But what happens? You have a high technical variation. Even within the same hospital, you have nurses that come and go, people that process with the different hands, with a different way, machines that change. So there is this problem, which is here. The variation is more embedded into the nature of the data, but here is technical and it's as difficult. The other thing which is difficult is that you often don't have replicates of the same patient. So if anything goes wrong, that is lost. And also, this opens the problem of the complex data handling and storage, which is a big nightmare when dealing with human samples. And Marcelo will talk something about it tomorrow. The other thing that, in particular with human, it's very, very difficult apart from cancer to get biopsies. So you definitely cannot get biopsy of the heart. So you have to use blood, which is the most accessible tissue that you can have from the human, from a healthy person or live person. So there are all this data that I will show you in a moment, all the analysis was done on RNA extracted from whole blood. And I can show you that was a challenge per se. There are many downsides, which I listed here, as well as upsides. Let's start with the up first. It's easy accessible. You can just bleed the patients like a blood test. Has a very wide dynamic interaction in the whole body, so it tells you of the whole system, what's happening of the whole system. But this is a downside as well, because it means that it contains a lot of other information that is not specific to what you want. You can extract the RNA, microRNA, and DNA very easily from blood, relatively easy from blood, and it can be done from the same sample, which is good, great. Can be stored in biorepository. Is a very complex tissue, but rich in information, and fantastic diagnostic tool, because you just can look at the markers in your blood. But what are the downsides? For me, the biggest downside of all is a lack of specificity, because it goes everywhere, it contains everything, and the signals are so convoluted that at times it's impossible to actually deconvolute them. There is a large amount of hemoglobin. If you use fluorescence assay like microarrays, that's a problem, because it binds to the biotin and enhances the fluorescence, so the background levels are very high. If you use sequencing, you read a lot of hemoglobin RNA that you don't really want to read. So a waste of space, basically. And from the biology point of view, there is a lot of circulating microvesicles, there are apoptotic body, and circulating cells that don't belong to the actual thing that you are looking at. So what happens in the blood as well, it is very affected by what we call environmental confounding factors. Because, for example, if I go out of the door and I get scared, my blood profile changes quickly because I have a reaction. If I've been told to go to the hospital to get my blood taken without having breakfast, and I kind of care less, and I'm hungry and I eat a bun, then my blood profile changes. So we had to find a way to clean the data that we were collecting with tools that would preserve the underlying signaling. So this is a covariance matrix of blood samples, of RNA, microarray samples of blood of patients. And as you can see, and this is, in theory here, there should be at least 3 groups, but you can't see very much what's going on. So we used the tool developed in Neil's lab with Niccolo Fusi, who's now in the States, called Panama, to try to deconfound the data before we could do anything with it. And now you can start to see the blocks that are coming out, although this tool cleared most of the signal. So the signal is now very, very low. What you would really want to aim is something like this, where you have a very clear distinction between the groups that you are studying, the categories of patients that you are studying. So how did I choose my correct methods? I had to do transcriptome analysis on 30 patients on a disease that the whole world has and is a major killer for everyone. So I didn't have any other choice than trying to find something that, rather than trying to estimate accuracy of the gene expression, was better at estimating uncertainty. Because I thought if I do a good estimate of the noise of the uncertainty, then maybe I'm better off finding true positives. So I firmly believe this because, as I said, especially when you deal with clinical samples, you never have enough samples. So you're better off with a model that handles the noise and the uncertainty, the noise and the uncertainty really well. And then I carried this in a downstream analysis and trying to reduce the number of false positives and come up with some candidates that then could be tested. So I write this here and say good measure of uncertainty helps to denoise the data. In a way, yes, if we call noise everything else that is unknown, obscure, technical noise is easier to get rid of, but it's this other type of nonlinear noise which is totally unknown and difficult to measure even, that is the problem. And therefore, in this particular case, when we don't have the patient anymore and we cannot go back and retest, it's important because it you need confidence in the results. So I used this program, this model, which I've been working together with Magnus Rattray, who you heard previously in this week, which is able to infer the signal, the distribution of the true signal from microarrays from data that we observe and we measure with fluorescent intensity. There is no time here for explaining all the details, but what you see is something like this, particularly when you do the estimation of the gene expression for each one of these probe sets, or of genes or transcripts. It's not really transcript, they are genes. It is just a sum of gammas which are established in a closed form and are able to give you as an estimate of the distribution of the true signal. Then you can use the mean of that estimate and the uncertainty for it. This enables you to do something else as well, because as I told you, we did have only 33 patients, but when you then want to ask the question, what is changing between one and the other? If I use these famous p-values, which are my worst enemies, it really didn't work. So I had to find a way to define a single distribution for each of these categories. So I had 3 classes, 2 different types of heart attack and a non-heart attack. And they all, if I plotted them all here, the distribution for each one of these patients, they all looked like this. Now, considering that 2 classes are, in theory, the same disease because they all had a heart attack, and the 3rd class didn't, this is a pretty bad overlap. So what I did, you can't see this is a red one. So what we did, we used a Bayesian hierarchical model based stemming out of the Puma, where we actually combined this and trying to find the posterior distribution of what the model believed being the representative form of the class, of the characteristic of the class. But because this data was particularly difficult and noisy, but not technically noisy, we had to also filter. And we thought that, no, the green doesn't come out. I can't see it. We had to really think very clearly on how we were filtering this data. So we used the variance that we predicted through this model, the variance that we observed, and then we filtered by looking at the ones that were going below this ratio. So you can't see here, but this is what they looked at the beginning. The ones that are filtered, they look like here. And these were the ones that we threw away, which were all the ones that were negative or very, very low signal. One thing I have to say, these type of methods are very unstable with low signal because the way they are and because the fact that we never have enough samples to improve the variance at that very low signal. It's too close to the background noise. So after that, as a downstream analysis, what we did was to use then the mean of the classes that we predicted, or representative of these classes in this way, to create a probability of positive log ratio, and use this probability to define a false discovery rate. And this is how they look now, that at least they are separate and they don't overlap. Doing all this, which is listed here, so that's our pipeline. So we did the expression, the quantification of the expression values, we corrected for the confounding factor, we filtered the data, we did all the downstream targets, and then there was some validation. So we ended up with 93 genes across the 5 time points, and then we used different methods called real-time PCR, with tilde cards to validate only 44 of these, which were chosen to match the patients and their clinical parameters. This was to minimize as much as we could the interference of the environment, of the background of the patients. And we did some functional annotation. It doesn't matter what is written here, but these functions were all related to the process of cleaning out of the dead stuff that happened in the blood, necrosis, apoptosis, and also a lot of it was to do with inflammation. But what was striking is that there wasn't much in common across the time. Only one gene was common everywhere. So common, I mean across, it was kept dysregulated across the time up to 90 days. I have to say, for the one of you who don't know, at 30 days, a patient is officially discharged. So after a heart attack, at 30 days, a patient is cured. And we looked up to 90 days, and this profile, it was still kept. This is the only pathway that was enriched, and it is an inflammation pathway. This is how the validation looks like with a heat map, and you can see that there is now, you can distinguish between the infarct people and the no-infarct people. And also this is the validation with the PCR, which is not as clear-cut, but it gives you an idea that the trend is right. So these models did have, and this was accurate, was validated, was confirming the results for about 70% of the targets that we put, and it's very high. One thing that we also did is we had this list of genes, we had a complex tissue. I did it in silico, so I used the full blood count to try to find out what was going on, what was the major player of all these possible cells. But also what we did, we took five healthy volunteers from the Department of Biomedical of Cardiovascular Science, and we split the various components of the blood. We put it on the cards, and we tried to figure out whether these genes belonged to platelet, to white blood cells, or to other parts, neutrophils, for example. And as you can see here, you can't probably read it, this is the platelets, and this is the white blood cells. So there is clearly a component here that is suggesting that platelets play a big role as well as white blood cells. So what we learned is acute coronary syndrome, all about platelets. They play a role before, but also they play a big role after from what we're seeing in this data. We have identified huge weight on pathways that are connected to inflammatory responses. Can this be modulated by the platelets? Do platelets play a role together, the white blood cells, in modulating the inflammation and therefore putting the patient at risk of a second one? And these are still questions that need to be brought back to the clinic and do further study. The last project that I want to sort of share with you is more to do with stem cell, and it's very, very new. It's just happening. We have in Sheffield established a single cell facility. We have the machine, we have the lab, and we are just starting this based on the experience of a stem cell center that has been established in Sheffield now for about 10 years more. So the project is about stem cell therapy and the use of stem cells in living individual, stem cells specifically derived to treat or prevent a disease. So we're talking about someone who's becoming deaf, and we use stem cells to replace the dead cell, or to hope to replace the dead cell so that that person can hear again. There are various points that still need to big time to be addressed. The first one, we use what is called human pluripotent stem cells. They come from embryos that are donated from the IVF clinic. And these cells, before they can be even conceived to be treated for any type of therapeutical intervention, they go through a long process which is called clinical grading process. So these cells will have to be clinical graded, which means they'll have to be clean, and they'll have to have all sorts of, there is a book of regulations that they have to comply. But what happens, even when they comply with all this regulation, is that the pluripotent stem cells, the human pluripotent stem cells, in culture acquire mutation. These mutations, various mutations, these mutations can also be, they propagate, so the cells, the cells with the mutation proliferate more than the cells without the mutations. You can use inhibitors and specific other protocols to improve this effect, but it still does happen. And this means that if you put a stem cell into the body of someone and this stem cell has a mutation and then it gets mad, then you have a cancer. So this banned totally the use of stem cells that have mutations for cell therapy. We don't know what are the mechanisms of this phenomenon that is called selective advantage, but it will be useful to try to model and investigate what are possible methods that we can use to suppress this. Because it doesn't matter that you have one cell with a mutation, but it does matter if you have the whole culture taken over by this mutation. The last point is that the problem is that to go ahead and have the approval for the clinical trials, Marcelo will say something more about that tomorrow, the cells needs to be absent of any copy number variation, which means that it's almost unachievable. And very, very difficult to obtain, and these limitations clearly stops us to advance in the field of cell therapy. So there are two major questions that we are trying to study. First one is how we predict the occurrence of variants and minimize them, and this is still unknown, and how we can identify functional significant genetic and epigenetic variants, because you can have a mutation, but that mutation can be there, useless. We have lots of mutations as I speak, things going wrong, but others have a functional effect. So how do we identify these? And how can we make use of the information that we have here and the identification of functional significance for translational on individual patients? So very, very quickly, just to give you an idea of what I meant, is when you have, the green is terrible here, I can't see. When you have a culture of stem cells spontaneously, and it's not known how this happens, one cell acquires a mutation. And there is a process that is called a mutation process. Then it triggers in a selection problem. So the cell with the mutation takes over the culture, and that's the problem. So what we want, and clearly this means that we have consequencing for the manufacturing from the biological, but also we have consequences when we want to put these cells into clinical use. So how we are trying to study this? The first thing that we're trying to do is, okay, let's try to estimate what is the mutation rate of this. That we don't know how it happens, but at least we can try to estimate what's the mutation rate. So somehow we can try to control how many times we passage this cell from one phase to another. And we are doing this in collaboration with the Stem Cell Center in Sheffield and the Sanger by looking at whole genome sequencing and bisulfite sequencing for 20 clones that come from a population of stem cells. They are expanding in doublings, about 90 of them, and sequenced, cloned and sequenced. So it's an enormous amount of data that will have to be integrated between the genomic and also the epigenomic. And what I hope, we have RNA from this as well, and I hope that we will have enough resources to actually do RNA, and that we have the problem then to integrate all this. And hopefully evaluate the mutation rate in relation to epigenetics, because it does happen in culture, remember, so something must happen biophysically onto these cells that triggers the mutations. The second question is, I put one up there, it should be two. The second question is a little bit harder to answer. The way we are approaching it is by looking globally at what's going on in our genome. There is this project, International Stem Cell Initiative, which was published, I think, 2012, studied about 160 different cell lines across the world and compared their genomics to see where mutations were occurring more frequently in the genome. You see here all the chromosomes, and all these bars are mutations that have been identified in the cells. And I don't need to go into details, but it's easy for you to see that there are some chromosomes that seem to be more prone to mutations than others. And therefore, it was worth to trying to extrapolate some sort of a prior knowledge on this that is really embedded in the way our genome is, and probably stems out for the evolution of it. So what we are trying to do is we're trying to collect, using SNP bit chip arrays, all the information that we have on the SNPs that are available that are present in these cell lines that we can have hold of, and evaluate whether there are copy number variation or loss of heterozygosity. And sorry. The SNP arrays contain about 2.3 million of markers, and normally you evaluate them by looking at these two quantities, the B allele frequency, which is the contribution of the B allele in the SNPs. So so you'll have the A allele, because B will have 0 contribution here. You'll have the AB, the 50-50, and then you'll have the B allele up here. If everything is fine, this looks a line with a little bit of noise, but all straight. When something goes wrong, there is a gap. Now, we don't know if gap is an endogenous loss of heterozygosity. Therefore, we look at the log ratio, which is the log ratio of what is predicted and what is expected. What is predicted and what is observed. If everything is balanced, is around 0, and there is a little noise between 0.5 and minus 0.5. If there is a dip or an enhancement, then you call it a copy number variation. It's very noisy, it's very difficult. This data is very unclear and very unclean. But what you can see in our data, which don't contain copy number variation as such, you see that areas that I called noisy. But these areas are actually in correspondence of those chromosomes in that map that I showed you before that seem to be acquiring more mutations. So we are developing some methods to combine the prior knowledge that we have on the whole genome, on the map from the ISKI study, what we can see at each single line that comes from a person, it's a fertilized embryo. So to identify areas, the potential hotspots where mutation can occur, and put that together with knowledge of expression and epigenetics. So in order to study the possibility of associating functional importance, we are trying to use the development of deotic stem cells in 3D culture, and therefore trying to change the environment and hoping to trigger by epigenetic changes to try to identify these functional impacts at system level. We have to integrate all the data, and we definitely need to build a predictive model for selection. But what is clear is that we certainly need to work at single cell level. If you look down here, what I've reported here is data from Oliver Thompson in Stem Cell Center, which he had 3 different lines, 14 I think are here, coming from one line called S6 and another line called S14. These are the absence and the presence of a stem cell marker of pluripotency. And a set of markers that are well-known stem cell markers. And he also annotated here what do they belong to. What you can see is the enormous heterogeneity of these cells that are supposed to share the same markers. They are stem cells, they're the same line, and they are at the very initial phase of it. They are pluripotent. So, for example, this is one that is quite clear. So look at this one, and this particular markers for this line, it's definitely all over the place, and it doesn't follow the pattern, and so this should be all red. But also, for example, this one, GATA6, it's very clearly not there. There should be a positive. So the reason that I'm saying this is because the stem cells are such an heterogeneous culture. In order to be able to take this and put it into a patient, we really need to make sure that each single cell is right. And so now it comes the single cell that you heard in these days quite extensively. So because I'm finishing my time and talking too long, I just want to briefly give you a summary of this. So I hope that I've convinced you that although modern biology studies all the fine details and large scale, we still have to use different disciplines to integrate all this at the system level approach. It's true that the darkness of the microscope room is over, but sometimes it's good to go back in there to make sure that what we're doing is right. We have very large data sets in assay, and what we also have is a huge amount of uncertainty that comes with this data. And then we have a need to quantify that so that we can estimate the proportion of the false discovery for the selection of our targets. Definitely, from what I told you, lack of sample size, very difficult, complex situations, tools from machine learning definitely have a huge impact in advancing this data analysis. And finally, when we go to model that have a very, very high uncertainty and the signal is so low, like a single cell analysis where the signal is so very close to the noise and it's so very difficult. So important is more the success of using any machine learning techniques and methods stays in the fact on how well you are able to model the uncertainty, because that will enable you to dissect it out from the true signal and hopefully enhance what is the signal in the background. I just want to close to say that this is what I believe is the road to personalized medicine. We started with computational models, we go to the bench, and then we go back to the computational model, and then we're ready to go to the bedside. And as all interdisciplinary scientists, my acknowledgement, suffer the curse of dimensionality. You'll see all the people that helps in all the stuff that I've shown you there. And this is the MRC platform, the UK Regenerative Medicine platform, which is called Pluripotent Stem Cell Platform, that is conducting all the stem cells. But mainly, and all the people here that you can read, the Stem Cell Center, the British Heart Foundation. But I think we need to thank the patients and all the volunteers that were really running away from me every time they see me because I wanted some blood from them. And you to listen to me. Thank you.